Come on. At least name of a single one ganglion. Huh? Do you know supraesophageal ganglia?
Yes, sir.
Yes. Then it is followed by subesophageal ganglia, and it is connected by circumesophageal what? Commissure or connective? I have also told you the difference between connective and commissure. What is the difference between commissure and connective? See, I am using the words circumesophageal commissure, circumesophageal connective, so to say. So, when the word commissure is used and when the word connective is used. Do you know? Do you know the answer? Say yes or no.
No.
When two similar ganglia are connected, then we are using the word commissure. But when two different ganglia are connected, then we use the word connective. So suprapharyngeal ganglia, it's a fusion of two, is connected to subesophageal ganglia, a different ganglia. So the word here is circumesophageal connective. What is the name of the large ganglia which is found or which is formed by the fusion of the thoracic segment?
Ventral thoracic ganglion
Very good. Ventral thoracic ganglia. Now, what is the peculiarity of this ventral thoracic ganglia?
is formed by 11 pairs of ganglion.
Yeah, yeah. But still, what is its uniqueness is there in this ganglia?
It is formed by fusion of 11 pair of ganglion.
Of course, I agree with you that it is formed by the fusion of the 11 different segments in the thoracic region. But how it's having its uniqueness, and that is expected from you. Is it a solid or it is having an aperture? And what passes through that aperture? The blood vessels pass through that aperture. Which is that blood vessel? While studying the blood vascular system, we have come across these words. Okay. How many abdominal ganglia are present? How many abdominal ganglia are present? How many abdominal segments are there? Yes, Nikita, answer my question. How many abdominal ganglia are there? Or what are the different sense organs that we have studied last time?
Eyes, statocyst, tangoreceptor, and chemoreceptor.
Yeah. Eyes, it is compound eye. What is the functional unit of the compound eye?
Omatida.
Very good. Ommatidia. And we have also discussed how the process of image formation takes place in dim light and also in bright light. What is the role of statocyst? What is the role of tango receptors and so on. Now today we are going to proceed further and we are going to study the topic which belongs to your unit 2 section B, that is prawn culture, and just go through the points that we are going to discuss in the today's lecture. And this is the synopsis. The introduction, selection of the species. Of course, we are going to study the freshwater prawn culture, life cycle of prawns, freshwater prawn. What are the basic requirements for the prawn culture? How seed is selected? What are the different culture practices? What we need to, how the feeding process is continued, how the fertilization of the pond is brought about, what is the production, how it is harvested, and how it is marketed. So these are the points that we are going to study in today's lecture. So one by one we shall be proceeding here. Then what the first point is the introduction. Prawn culture has great attention because of its increasing demand in the international market. It has high market value due to delicious taste and high nutritive value. You know, the prawns, these are highly proteinous, nutritious, and delicious seafood, and which is high demand in the local and as well as the markets in the inland markets as well as in the local markets. Freshwater prawns, as you have we have studied prawn as a type. That's all of you are quite aware that it is an crustacean of having an economic importance, and which is also commonly called as shellfish because of its body is covered with the exoskeleton, hard exoskeleton, so also referred as shellfish. So culture of this freshwater pond is also called as shellfish culture. Now, selection of the species. Now, as far as freshwater uh, prawn culture is concerned, these are the two main species which are taken into consideration or given weightage or they are preferred while uh, going for the shellfish farming or shellfish culture. One is the Macrobrachium rosenbergi and another is Macrobrachium malcolmsoni, which we have studied it in detail as a type study. So how this appears? See, this is the species. This is the male and female of the Macrobrachium dimorphism. How to make out whether the prawn is male or female by observing its sexual characteristics. So can you tell me sexual dimorphic characteristics by which we can make out whether the prawn is male or female? Yes. We have studied Macrobrachium malcolmsoni. Or this type of sexual dimorphic characters are applicable to the genus Macrobrachium. Can you tell me the difference? Yes, Nikita.
Let leg is large in male.
Which one?
Second chillet.
Second chelate leg is huge, large in case of male. In both the sexes, you will find first and second legs are chelate, but the second leg of male, second chelate leg of male is very enormous, huge, and large, and that is what we can make out the difference whether the prawn is male or female. As far as this Macrobrachium rosenberg is concerned, it is mostly most preferred freshwater prawn, and it is found in the rivers, estuaries, and the brackish waters. It was introduced from Malaysia to Hawaii in the USA, Japan, Thailand, and other countries of Southeast Asia. But in India, it is found in West Bengal, Kerala, Orissa, Maharashtra, Gujarat, Andhra Pradesh in sizable population. So, in the riverine systems of these different states, you will find the amount or the abundance of this Microbrachium rosenbergi. It attains the size of 150 to 250 mm in length is the size, but maximum size attained, maximum growth is recorded about 320 mm in length. Now, while studying the external morphology, habits, and habitats of the Macrobrachium genus Malcusoni, we have studied that this generic genus of the prawn, it it is bottom dwelling and sexes are separate, no doubt. It attains the maturity about in about 4 to 5 months. Males are larger than female. As I have mentioned just now, second walking leg of male is larger, hence the name macrobrachium. Macro means large, and the word brachium, it is concerned with the arm. So, the name of the genus is macrobrachium. What is the feeding habit of the prawn? What you can say about the feeding habit of prawn? What type of food the prawn prefers?
Omnivorous.
It is omnivorous. Very good. It feeds on rotifers. It feeds on Noplia of the crustaceans, tubifex worms, fish eggs, and so on. Now, very interesting part to be noted here is that this species, this they migrate from freshwater to estuaries for breeding. See, they show migration for breeding from freshwaters to the from the rivers to the estuaries. And you know the estuaries is a transition zone, and it is an ecotone between two different habitats, freshwater and marine water habitat. isn't it?
Yes, yes.
Now they breed in the area having 5 to 20 ppm salinity. Now this is the very moderate salinity. It is neither high as that of the seawater, and it is neither low as that of the fresh water. So moderate salinity is there in the estuaries, which is preferred as a breeding ground. After the young ones, now they go there, they mate, they lay the eggs, eggs are fertilized, young ones, they come out of the eggs, metamorphosis takes place. But when the young ones grow up to the size of 2 to 3 centimeters in size, they start upward migration. Upward migration means from estuaries to the freshwater, where they grow up to the adult size. So this is the species. Another species is Macrobrachium malcolmsoni. Now this Macrobrachium malcolmsoni is a species which we have studied it in details. Now, this is also preferred because of these qualities, or you can say both of these species. You can find uh, they are they are having a different high qualities. First is high growth rate. Second, omnivorous feeding habit. High nutritive value, rich flavor, palatable, very good taste. That is why it is having a high market value. Scope of earning due to high demand in the market. So economic value is high of this species. Now, next point that we are going to study is the life cycle. During breeding season, as I've mentioned, that it is migrating from the fresh water to the estuarine water, and the breeding season is from May, June, and July. These are the months of the for breeding. Mature male and female, they copulate. Female produces about 40,000 to 1,50,000 eggs. So such a average egg laid by a single female is about 70,000, of zero point 0.6 to zero point 0.7 in diameter. The male deposits the chromatophore, mating occurs, female deposits, male releases the spermatophores, the eggs are fertilized by the sperms. The fertilized eggs are now attached to the pleopods of the female. Now, eggs, we have studied where these pleopods are located. Can you tell me where these pleopods are located? Yes. Where these pleopods are located?
Abdominal region.
Yeah, in the abdominal region. They are used in the for what purpose? For swimming purpose. So the pleopods, both the pleopods of the opposite sides, they form a bridge, and this is bridge. I will show you how these are hold. Yes. See, now these are the eggs. How these eggs are hold by the female in between the pleopods. And now after 3 weeks, after three, this egg fertilizes. They are attached to the pleopods. The eggs are now very bright red in color. After about 3 weeks, the color becomes, it appears to be salty. The eggs hatch to the larva after 3 weeks. See, egg hatch into grayish larvae. Now, after this, the hatchlings are grayish in color and the larva passes through 11 larval stages, and it takes about 28 to 37 days, and then it is converted into post larval stage, or it's a juvenile stage which remains at the bottom, and they now start this post larval stage now starts upward migration, that is migration towards the fresh water, and about 4 to 6 months it is transformed into an adult. But what are these 11 different larval stages through which the prawn passes? And these are these 11 different stages, you can say. First, I hope that during your zoology lectures, have you studied the crustacean larvae? I hope that you have studied. It's a general topic. Crustacean larvae. Nikita, झालंय का?
No idea, sir.
Okay. So this is how different. Every crustacean during its life cycle has to pass the certain stages of the larval stages, and very first larval stage from which every crustacean has to pass is the nauplius larva. And this is the Nauplius larva, how it appears. So it contributes, Nauplius larva, it contributes the content of the zooplanktons present in the water body. And how this is transformed into second and third Nauplius larva, fourth, fifth, and sixth. The, as the time proceeds, the size of this larva goes on increasing, and then after it is now gets transformed into first protozoa larva. Now you will find there is a bit size change in the size of this larva. This is about zero point 0.2 mm and this is about 0.5 mm, and gradually it goes on increasing from first protozoa, second protozoa, and third protozoa larva. The primitive stage of the zoea larva, which is also a crustacean larva. Then it passes through the stage called as mysis. First mysis, second mysis, and this is third mysis stage, or what we refer it as a post-larval stage. I hope if you have performed this practical last time during the fisheries, while we have had a practical as a mountings of the zooplanktons, and in that we have mounted different types of zooplanktons. Among these zooplanktons, mysis was one of the zooplanktons that we have observed it under microscopes. Other two zooplanktons were cyclops,
Euglena.
correct? Daphnia was also there, and
Def-
you have observed this larval stage, which is called as zoea larval stage, and we have also mounted this stage while mounting the zooplanktons while performing the practicals in fisheries BC part 2. Okay, so these are the different larval stages in which the prawn has to pass the life cycle, or it has to undergo the metamorphic changes. And as soon as it attains this zoea larva stage, the post larval stage, what it is commonly called as, and this is that stage when the zoea larva starts upward migrating journey towards the fresh water, and slowly, gradually it attains the maturity and it is converted into an adult. Okay, so next point that we are going to study is what are the basic requirements for successive prawn culture. Very first, that is there should be a ample amount of seed should be available. Unless and until proper amount of seed is available, you can't go for the freshwater prawn culture. Existence of suitable temperature, and this 20th temperature is minimum 28 degrees centigrade is the requirement for the proper growth or optimum growth of these prawns in the freshwater. Containment of suitable availability of water in the rearing tanks. Now, of course, prawns they live in riverine systems. Their natural home is the riverine system, but we can introduce these freshwater ponds in the pond culture practice also in prawn. So, what is needed here that as whatever the aquacultural practices, it may be a shellfish or maybe a finfish. The basic requirement for to carry out this culture practices, water is required, and the perennial source of water should be available for the practice of this culture of this shellfish. Provision of suitable type of feed for conversion to body proteins. Suitable type of food as a fish, as this shellfish is omnivorous. The food or whatever feeding we are going to supply, that feeding should be uh, that feeding should be of quality, that it should be a protein rich and that can be supplied thoroughly, and which will help in conversion or converting into a proper biomass, which is in turn should have a high protein value. So when you are having these basic requirements, and if you are going to fulfill all these basic requirements, you'll find, and it is sure that you are going to succeed in the uh, prawn culture practice. Now, next point here is seed collection. Seed collection from the wild areas. How the seed is collected? Wild means from the nature. Now, larvae and post larvae are obtained by two methods, and the first one is the collection of seed from wild. Means the prawn seeds are can be collected from the estuaries and the brackish water, because whatever breeding takes place and that breeding occurs in the, excuse me. Hi. अच्छा जी. Oh. Why we need to go to the brackish water and the estuarine water? Because as we have mentioned that prawns for breeding purpose they are moving from the fresh waters towards the estuarine waters, and up there the breeding their mating occurs, and after the mating you will find the as these sperms are spermatophores are released, and this spermatophores fertilizes the eggs externally, and the eggs are then accumulated, they are collected by the female in between the pleopods. We have observed the slide exhibiting the collection or the how the female is handling the eggs. So we need to go to the brackish waters, and with the help of fine mesh cloth bags which are having 2 meters, which are 2 meters long and 1 meter in diameter. See, this is the slide which is exhibiting that these women they are engaged in collecting the seed from the wild, that is from the brackish waters, and how they are employed. They are using and making use of the device for collecting the seed. Now this seed may be in the form of larva, maybe in the form of Nauplius larva, maybe in the form of uh, protozoa larva, or it may be in the form of zoa larva. All such type of seeds they are collected, they are sorted, and they are kept or they are sent to the different ponds as far as the scrutiny is done. And these are the some of the pictures of the post larval stage. This is prawns, Zuya larva, and this is you can see how tiny creatures are there, and this is bit enlarged larva, how they appear in the as a prawn seed as a whole. Next, see. Next is by collection of seed from the brood females. Collection of seed from the brood female. That means directly here the brood females are captured. These brood females are captured, and these brood females, as I have mentioned, that the brood females are carrying the eggs in between the pleopods. So these females are then collected from the brackish waters or estuarine waters. Three such females are introduced into the rectangular tank, which is called as a larval tank, containing 1% brackish water added with the formalin about 15 to 20 ppm. Now, egg hatch out in about 4 to 5 days. 50% of water is removed after 3 days and fresh water is added. That means slowly, why this change is made? Because slowly, we are going to make the environment which is suitable, which is similar to that of the fresh water, and the water is removed, and then the tank is well aerated, and the pH is maintained between 7 to 8.5. Now the larvae are fed with the नॉप्लिया. आणि हे पोस्ट लार्वल असेल तर नॉपलेस लार्वा आहे त्यांचं अन्न. फिश एग स्मॅश केलेलं बारीक केलेलं फिश एग, chopped earthworms, and about after 28 to 37 days they are now called as post larvae. That means they are now have attained the size of Zuya larva, or they are transformed now into Zuya larva, so called the post larva. They are stocked in the concrete tanks called as holding tanks at the rate of 5,000 per meter square. After 1 month, they are transferred to the production ponds in tins and polythene bags. Tins and polythene bags. So this is how the seed is collected from the broods. It is processed and then it is transformed. Now, next point here is the culture practices. Different cultural practices are carried out, and the first one is the pond culture, and this is the actual picture exhibiting the ponds in the field, how these ponds are constructed, and the prawns are reared in these ponds. Now, ponds used for the pond culture are called as production ponds because the post-larval stages of the uh, prawns, they are released here, and they are about zero point 0.2 to 1 hectare in size. The depth should be less than 1 meter. The inlet and outlet are screened by fine mesh nylon so that there should not be entry of the other predatory organisms, fish especially, which feeds on the larva of the prawn. Small number of Ipomia plants are reared to give shelter to the growing ponds. The ponds are fertilized with 200 kg per hectare of cow dung and 25 kg per hectare of superphosphate. Now these are the fertilizers which are applicable, which are applied. Why they are applied? Why they are applied? Why this
for
fer? Yes,
for growing algae.
for growing. Yes, phytoplanktons as well as zooplanktons. Phytoplankton is grown there, and phytoplankton supports the zooplankton. That's why the growth of the zooplankton is also supported. And these both of these, the algae as well as the zooplanktons, which grow because of these fertilizers, forms the food of the growing plant, because they are in the stage of post larva. So this plankton forms a primary food for them. So about in 1 month, larvae are stocked in these ponds at the rate of 25,000 to 30,000 post larvae per hectare. They are fed with supplementary feeds such as rice, vegetables, animal wastes, trash fish, mollusks, pig dung, and prawn waste. See, prawn waste again it is can be made use as a food for the prawn at the rate of 5 to 10% of the body weight. It depends on what is the size of the larvae which are growing in that particular water or particular pond once in a 3 days. The production rate is about 2 tons per hectare in 6 months. So if you are going for the pond culture. Our next practice is the polyculture. See, polyculture. Now it is a culture of prawn with the Indian major carps or other carps with rohu, katla, grass carp, silver carp, milkfish, mullet, tilapia. Along with this, the prawn culture is also carried out. The fecal matter of this fish is a good source of food for the prawns. See, the advantage is taken of this polyculture. In polyculture, the yield of prawn is less compared to the finfish yield. See, what is the yield here as compared to the fish? In polyculture, the fish yield is about 2 tons per hectare per year, while the prawn is about zero point 0.5 tons per hectare per year. Okay. So another method is the batch culture. Now, in batch culture, what is called the pond is stocked twice in a year, and two crops are made. The pond is stocked with the post larvae, and after 6 months, the entire prawn crop is harvested, and ponds are dried, drained, and again they are prepared for the purpose of stocking. And then the second batch of post larvae is stocked and harvested after 6 months. So after every 6 months, so in a year we can have two batches of the prawn culture, and it depends on what and how availability of the ponds, availability of the area, and availability of the water resources, and then and only then such type of uh, batch culture is followed. Next is the continuous culture. The pond is used for the continuous culture for about 1 to 5 years. Harvesting is done continuously for every 4 to 9 months, and the post larvae are added periodically after every harvest without draining, without drying. Such type of practice is done. The ponds are drained and dried after 5 years. Batch culture and ह्याच्यातला फरक, what is the difference here? In the batch culture, within a single year, the pond is dried and drained after every 6 months. Drained after every 6 months. But here in the continuous culture, the ponds are dried and drained after 5 years. After 5 years. During harvesting, large mesh sign net is used, and only largest prawns are collected. Sign netting is done once or twice in a month. In continuous culture, large and more uniform prawns are produced, and the production ranges for about 2000 to 4000 kg per hectare per year. As the mesh size is larger, only the full grown prawns are harvested, and rest of the young ones stages which are in the process of growth, they are left behind to grow properly, and then they are harvested. The next is the sewage fed prawn culture. Prawns are cultured here in the paddy fields filled with the sewage water. भात शेतीमध्ये. I think the topic integrated fish culture is going on, isn't it?
Yes, sir.
Yes. What type of practices you have studied in the integrated fish farming?
Sewage fed farming, duckry fish farming, pig and fish farming, cattle fish farming.
Okay, okay. Paddy field or rice field farming.
So also paddy rice.
Yeah, yeah. So prawns are also cultivated or they are also cultured in these paddy fields. In these fields, the yield of prawn was high as compared to that paid with the normal waters. So it has been observed that if it is cultured in the paddy fields, it supports or it accelerates the growth of the. Then next is the Pokali culture. Now this Pokali culture is a traditional culture which is followed or which is practiced in Kerala. The prawns are cultured after the paddy harvest, भात कापणी झाल्यानंतर harvest in the low saline period. After harvest, the prawns are stocked and reared for about 4 months with the supplementary feeding. So even though the fish, this shellfish farm, even though they are having small patch of land in the vicinity of their houses, these people in Kerala, they are practicing such type of culture. Okay, so these are the different culture practices which are performed for the prawn culture. See, this is the picture exhibiting the culture in the paddy fields. Next is the feeding. Now, prawns are fed on living organisms, vegetable matters, dead organic matter, and debris. Artificial feed is given in the form of thrash fish. थ्रॅश फिश म्हणजे मच्छी मार्केटमधलं जे काय टाकाऊ राहिलेलं असेल, फिन्स साफ करून जे राहिलेलं असेल, ऑल दॅट इज द थ्रॅश फिश or the fish which are brought along with the catch, but they are not having the market value. Such tiny fish which are thrown, then that fish also is referred as a thrash fish and can be utilized as a feed for the prawn culture practices. Mollusks, pig dung, broken rice, fruits, animal farm waste, prawn waste, all these are used as a prawn feed and they are artificially fed. So artificial feed is given once in 3 days at the rate of 5 to 10% of the body weight. Next is the fertilization. As I mentioned, the cow dung, the organic manures as well as the inorganic manures, fertilizers, they are used for the process of fertilization. Cow dung is about 200 kg per hectare and a superphosphate of 25 kg per hectare is used as a fertilization. The very intention of the fertilization is to accelerate the growth of the planktons in the water body. Production. Freshwater prawn grows to the maximum length of 32 centimeters and weighs about up to 20 grams. So this is the size or the high weight and the length of the individual prawn in the pond. The production rate in the pond culture is about 4,000 kg per hectare per year. In the polyculture, it is about zero point 0.5 tons per hectare per year, and in continuous culture, it is about ah, okay, 2,000 to 4,000 kg per hectare per year. Next point is harvesting. Harvesting is done by pulling a seine net. Care should be taken to be sure that the seine net reaches the bottom of the pond because large males may avoid the net by burrowing into circular breeding depressions built. And last point here is the marketing. Freshwater ponds pond can be harvested where there is when there is high demand and can be sold alive. They can be processed and stored due to high demand both inside the country as well as it has having export markets abroad. So either they are sold live directly in the markets or they are processed in the form of tin foods and they are exported in the international markets. See how they are harvested. Huh? You can see the net in which these prawns are harvested and they are markets. See, and how they are sold in the market. Okay, uh, so you are having practicals now, and we will stop here. Okay.
Yes, sir.
हॅलो हॅलो अरे ऐकायला येते का हॅलो
हाँ येते मॅम
येते ना लवकर लवकर जॉईन व्हा आतापर्यंत 8च आहेत फक्त ऐश्वर्या पिसे तेजश्री माने स्वप्नाली गायकवाड बागवान विपुल घोरपडे वैष्णवी today's practical which is related to the comparative study of or comparative account of brain. We complete this topic in theory, but also this topic is also in our practical, and brains are identified, described in practical study, or identified and sketch and label. In practical, there is a theory related practical. Theory, comparative account of brain, in that related to this comparative study of brain, fish, amphibia, reptile, aves, and mammal. So these are the 5 comparative study animals. So how this brain, it is the main part and it is differentiated according to the habit and their behavior. Brain is the important part which is covered by brain box, or it is also called cranium. Inside the cranium, there is also meninges. So in fish, meninges single layer, so it is called meninx. And other animals, starts from the amphibia to the mammal, these are hard covering, it is called as a cranium or brain box. So how brain is, how this neural, first one is the neural tube. This neural tube, how it is differentiate into the three part, how brain divided into three part, from the protochordate up, brain is not well developed, but स्क्रीन दिसते का रे? सर्वांना स्क्रीन दिसते का? पीपीटी दिसते का?
नाही मॅडम.
दिसते ना? हा. So
आता दिसली मॅम, दिसत होती आता मॅम.
दिसत होती ना? थांबा आता परत दाखवू.
हा दिसली मॅम.
दिसते ना?
हा.
सो डेव्हलपमेंट ऑफ द ब्रेन, आता हे थिअरीला पण तुम्हाला झालेलं आहे. ब्रेन इट इज डिवायडेड इंटू दी three major divisions: prosencephalon or forebrain, mesencephalon or midbrain, and rhombencephalon or hindbrain. So these three main part, it is divided. This is the neural tube, embryonic neural tube. This blunt part it is called as the encephalon, and lower part it is called as the spinal cord. This tube then divided by the constriction into the four animal and behavior of the animal. These again divided into the three part and differentiation. These are the differentiation. In the forebrain, presence of olfactory lobe. It is another name, renencephalon. So this is the another name or renencephalon, olfactory lobe, forebrain part. Then this brain, second part, it is called as the cerebrum or cerebral hemisphere. It is also called telencephalon. This around this cerebral hemisphere and all, these are the outer covering, it is called as a pallium. And the cerebral hemisphere, so these are the olfactory lobe, cerebral hemisphere, these are the second part of these two parts of the forebrain. Then third part is the diencephalon. So this is the diencephalon. Forebrain consisting olfactory lobe, cerebral hemisphere, and diencephalon. And on the upper surface of diencephalon, there is the presence of pineal body and parietal body. And lower surface of this, there is the presence of hypophysis and optic chiasma, cross of nerve, which is called as a optic chiasma. So these parts up to the diencephalon included into the forebrain. And these names are also different. These are asked on the MCQ. Olfactory lobe is also called dash dash renencephalon. Cerebrum is also called telencephalon. Then diencephalon is also called thalamencephalon. So these are the pineal body and parietal body. These are the stalk, pineal stalk and parietal stalk. Then this is the middle part of the brain or mesencephalon. It consisting optic lobes and crura cerebra or crus cerebral nervous tissue band. It is called as a crura cerebra. And this is the optic lobe. This part is called as a midbrain. And hindbrain on rhombencephalon, it consisting the cerebellum, or it is also called metencephalon, and medulla oblongata, it is called myelencephalon. And this together, these are included into the rhombencephalon or hindbrain. So these are the differentiation of the brain, and these are the cavities of the brain. In the brain, there are cavities present. In the upper surface, there is a presence of first and second, it is called as the lateral ventricle. Then third, it is called diocese. Fourth one, metacese. And these ventricles connected to the one another. In this ventricle, there is a presence of cerebrospinal fluid. Cerebrospinal fluid. which protect brain from the mechanical shocks. So parts and name: olfactory lobe, renencephalon, cerebral hemisphere, telencephalon, and these are three included into the. Then midbrain consisting of, it is called mesencephalon, metencephalon, and myelencephalon, and it is called myelencephalon. So these are of the brain. Then the comparative account. So in the fish, first one is the fish. In fish, example is the elasmo branch, and name of this it is called as scolidon or dogfish. Name of, because of this the lobes, these are well developed, and these are locating the smelling or smell. So dogfish or scolidon, it is famous for smelling well developed the part. One of the blood drop in the where there is a present in that part, it firstly reach that blood drop. So powerful smelling in dogfish. So these olfactory lobes are well developed. So brain of this fish, it is divided into the forebrain, midbrain, and hindbrain. So this part it is called as a forebrain, which are the again divided. These are the olfactory lobe, olfactory peduncle, then cerebrum, and diencephalon, which is also covered by the optic lobes and cerebellum. So diencephalon is not seen in this diagram. First part is the olfactory lobe. It is also called telencephalon. It is called as a renencephalon. It is sac-like structure, and which is joins to the cerebrum. Cerebrum is undivided by a peduncle. It is called as a olfactory peduncle. These are the two olfactory lobe, and these are well developed, not found in the other animals or other vertebrates, only well developed in the this fish. So olfactory peduncle, these are laterally joined to the cerebrum. Cavity of this rhinocele, this the olfactory lobe cavity, it is called as a rhinocele. Then rhinocele, the peduncle it joins to the cerebrum. Cerebrum, it is undivided rectangular part, and this it is called as the cerebrum. When these are equally divided, it is called as a cerebral hemisphere. This is the part pineal body and pineal stalk, and below them there is a presence of diencephalon, which is covered by cerebellum and also optic lobes. So this is the part, it is the dorsal roof of this diencephalon, it is called as the anterior choroid plexus. There are two anterior choroid plexus, one is the anterior, one is the posterior. One is the roof of diencephalon and one is the roof of medulla oblongata. So these two choroid plexus, anterior and posterior, secrete cerebrospinal fluid. Then the ventral surface of this diencephalon, there is a presence of cross of nerves. These nerves are called optic chiasm. Below them there is a presence of infundibulum. Around this there are two bodies. These are called inferior lobes and saxus fasculosus. And this is the hypophysis. Hypophysis infundibular, there is a together form pituitary body. So these are present only in the fish, not other vertebrates. Why? Because of this swimming activity, these parts are also well developed. Then, so this is the ventral part of the diencephalon. Then second part, midbrain. Midbrain consisting optic lobes. These are the two lateral side optic lobes covered by the cerebral cerebellum. And these optic lobes are sense of function, sense of sight. These then optic lobe and the cerebellum, third part of the hindbrain, it is the third part of the brain, and in that presence of cerebral hemisphere, cerebellum, and laterally there are outgrowth like structure, it is called as a corpora restiformia. So these are also important for the swimming activity, or there are these helpful, these restiform bodies, these are also helpful to the or assist to this cerebellum. Cerebellum is the well developed because of their animals swimming activity. Then fourth part, in hindbrain, there is a presence of another part. It is called as the medulla oblongata. Medulla oblongata dorsally broad and anterior it is broad and posteriorly narrow. So medulla oblongata, it controls the all necessary activity, respiration, uh, circulation. All activities are these are carried out by the medulla oblongata. Then cerebellum. In the cerebellum, there is a no cavity is present. It is called as the epicele. Epicele cavity. Only in this animal or fish, cavities are present. It is called as the epicele. So now we see the cavities are ventricles in the brain. These ventricles consisting fluid. This fluid it is called as a cerebrospinal fluid. So cavity of this olfactory lobe, it is called as the rhinocele. These are two sides, rhinocele. Then cavity of cerebrum, it is called as the lateral ventricle or paracele. So these two lateral ventricle unite together, it is called as the foramen of Monro. Then after that, cavity of diencephalon, it is called as the diocele or third ventricle. Optic lobes cavity, it is called as the optocele. And narrow canal in between this epicele and optocele, it is called as the eta. And cerebellum's cavity, it is called as the epicele. And medulla oblongata's cavity, it is called as a metacele. So these are the cavities of the brain. And this is the brain of fish. So it is also called elasmobranch. We see in this olfactory organs are enormous, large, attached to the cerebrum by olfactory tracts or peduncle. Optic lobes are also relatively moderate in size. Then saccus vasculosus, it is attached to the pituitary. Pineal apparatus, which is also well developed. Cerebellum is large due to the active swimming habit. And to help this, there is also corpora restiform bodies. So this is the first vertebrate. It is in the well-developed brain than the protocordes or cyclostomes. Next one is the amphibia. In amphibia, example, the well-known example is the frog. In frog also there is a brain is divided into the forebrain, midbrain. These are the midbrain and hindbrain. So this is the part hindbrain. In the forebrain, it consisting, there are olfactory lobes. These are anterior most part of the cerebral hemisphere. In this frog, there is a cerebral hemisphere. These are divided, equally divided. So it is called cerebral hemisphere. Elongated, tubular and globular posterior side, cerebral hemisphere. Olfactory lobes are not well developed. In fish, these are well developed, but in the animal frog, this animal is sluggish animal. So these are not or less developed olfactory lobes. Cerebral hemisphere, these are well developed because of this hibernation, estivation condition and instinctive behavior of this animal. These are the also well developed. So cerebral hemisphere, these are outer covering, it is called as a pallium. Lower surface, it is called as a corpora striata. On the surface of the cerebral hemisphere, thin layer is present. It is called as a pallium. And lower surface, these are consisting. It is called as a corpora striata. So cerebral hemisphere controls the all activities in our control. So next part is the diencephalon. This is the pineal body. Parietal body is absent, but pineal body is present. Diencephalon, diamond-shaped structure. Diencephalon, which is covered by this cerebral hemisphere, and also there is a optic lobes. So this diencephalon, ventral surface is showing cross of nerve. It is called optic chiasma. Below them, there is a presence of infundibulum and pituitary. So these are saccus vasculosus, lobe inferior. These are absent because the animal is sluggish animal, and these are the organs only present infundibulum and pituitary. So these are the three parts in the forebrain. olfactory lobe, cerebral hemisphere, and diencephalon. Cerebral hemisphere, it is called rhinoceros. Then cavity of cerebral hemisphere, it is called as a lateral ventricle. These are together form foramen of Monro. So cerebral hemisphere cavity, it is called as a lateral ventricle. These together form union, it is called as a foramen of Monro. Then diencephalon cavity, it is called as a diocele or third ventricle. Then forebrain consisting two optic lobes, and also there are crura cerebra, nervous tissue band present lower surface of the brain. It is called as a crura cerebra. In this side, starts from the midbrain up to the last. These are the crura cerebra, or it is also called crus cerebra. Then these optic lobes are lateral side. Number is 2, and these are also not well developed. Sense of sight. Function is sense of sight. Then crura cerebra, second part of the midbrain, it is called as the crura cerebra, nervous tissue band. Then hindbrain consisting medulla oblongata, it is the uppermost part, broad and posterior narrow. This is the posterior choroid plexus. Roof of this medulla oblongata, it is called as a posterior choroid plexus, and which produces cerebrospinal fluid. Anterior choroid plexus on the roof of diencephalon, these are the anterior choroid plexus. And these two choroid plexus, these are produces cerebrospinal fluid. Then in between this optic lobes and also medulla oblongata, there is a diamond shaped structure, it is called as a cerebellum. Cerebellum also not small transverse band like structure, and it is not well developed, it is poorly developed. Because of this, animal is sluggish, it is poorly developed. And these are cavities of the brain. In the first, these are the right in the midbrain. This is the rhinocele olfactory lobe cavity, lateral ventricle, cerebral hemisphere cavity. There is a union, this it is called foramen of Monro. Then this is the diocele, diencephalon cavity, optic lobes cavity, it is called as the optocele. The narrow canal in between metacele and optocele, it is called as the ether or aqueduct of Sylvius. MCQ याच्यावरती विचारले जातात थेरीला तुम्हाला. What is the name of canal between the optocele and metacele? Narrow canal between optocele and metacele, it is called as the ether, or another name aqueduct of Sylvius. And fourth ventricle, or it is also called medulla oblongata cavity, called as a metacele. So this is the brain, not the only development in the cerebral hemisphere. It is highly well developed than the fish. Only development is the cerebral hemisphere. So this is the characters of the brain: small olfactory lobe, large optic lobes. Optic lobes are cooperating center in the amphibian brain. Then two cerebral hemisphere shows greater development in accordance with the more complex activities of locomotion, hibernation, breeding. Then poor development of cerebellum, it is transverse band like, so decrease the muscular activity. Medulla oblongata also small. Pineal body is present in all modern amphibians. So this is the brain of frog. Then we compare next part, or reptilian brain. For example, lizard brain, or it is also called Euromatrix. So in this, अरे ऐकायला येते ना? Hello.
Yes, ma'am.
हो मॅम येत आहे. हो मॅम.
काय ऐकायला येईना? बोला ना.
हॅलो मॅम.
हा बोल.
हा येतंय ऐकायला मॅम.
डायग्राम वगैरे दिसतात ना क्लिअर?
हो हो मॅम दिसतंय.
का हेच तुम्हाला थिअरीला पण आहे. याच्यावरती कुठलाही क्वेश्चन विचारला जातो. कम्पेअर विथ फिश ब्रेन टू दी अँफिबियन ब्रेन टू दी रेप्टाइल ब्रेन. कम्पेअर, हे रिव्हिजन अशी समजा तुम्ही आता. सो नाउ दिस ब्रेन, इट इज कॉल्ड एज ए रेप्टिलियन ब्रेन. इट इज ऑल्सो वेल advanced than the fish brain. So these are divided also forebrain, midbrain and hindbrain. So olfactory bulbs are present. These are present on the anterior surface, and there is the olfactory peduncles are also present in the forebrain. Olfactory bulb, these are attached to the cerebral hemisphere. Cerebral hemisphere, two larger oval bodies. In between them, there is a fissure, dorsal fissure is present. So these are two equally divided, so it is called as a cerebral hemisphere. The pineal body is also present, and this is the olfactory bulb, then olfactory peduncle, cerebral hemisphere, and third part of the forebrain, it is called as a diencephalon. It is compressed part, it is called as a diencephalon. So smaller and not well developed. So the lower or ventral surface of this diencephalon, there is a presence of this. There are optic chiasma, cross of nerves, optic nerves. It is called as a optic chiasma. Below them, there is a infundibulum and hypophysis. Together, these are called pituitary body. Then cavities of the brain. So this is the cavity, olfactory bulb. It is called as a rhinocele. So cerebral hemisphere cavity, it is called paracele or lateral ventricle. These unite together, it is called as a foramen of Monro. Then cavity of diencephalon, it is called diocele. Optic lobes cavity, it is called as optocele. Then this is the eater, narrow canal, it is called as the eater or aqueduct of Sylvius. And medulla oblongata cavity, it is called as a metacele. So these are the only olfactory bulbs are present, but in this also the brain is not well developed. Next one is the description we see in the, so it is advanced than the amphibian because of terrestrial mode of life. Telencephalon or cerebral hemisphere, it becomes largest. Then two olfactory lobes are connected, cerebral hemisphere by olfactory peduncle. Then parapineal body is called as parietal body. Only some of this modern lizard, there is the presence of parietal eye or parietal body. All animals, there is the presence of pineal body, but only in the reptiles, there is a presence of pineal body as also parietal body. So third ventricle or diocele, it is also reduced. Cerebellum is pear-shaped, larger than the amphibian. So this is the cerebellum, triangular shaped cerebellum. It is also larger than the amphibian, but this it is not more advanced like bird or mammal. Next one is the bird brain. So a common example is the pigeon or rock pigeon. So this is the well developed olfactory. These are the cerebral hemisphere. Olfactory lobes are poorly developed because they need no need to the smell. So these are the aerial animals, no need or related to the smell. So these olfactory lobes are poorly developed. So forebrain consisting olfactory lobe, cerebral hemisphere, and diencephalon. So diencephalon, and which is also covered by the cerebral hemisphere and also the two lateral optic lobes, and also cerebellum. This is covered by the diencephalon. The cerebellum, the cerebral hemisphere, it is well developed because of this instinctive behavior of the bird. All are aerial animals. So there is a, these are high degree of intelligence and instinctive behavior. Cerebral hemisphere, it is well developed. But there is a cavity is present, it is called as a lateral ventricle or paracele. So cerebral hemisphere, these are equally divided larger bodies, and these are well developed than the reptiles. Olfactory lobe, there is no need to the, these are poorly developed, these are the animals, aerial animals, so no need to this sense of smell. So this is the part. Then third part, it is called as the diencephalon, which is also completely covered by cerebral hemisphere. But ventral surface of the diencephalon, there is a presence of the cross of nerve, which is called as the optic chiasma. Below them, there is a presence of the infundibulum and also hypophysis. These are the infundibulum and hypophysis. Then midbrain consisting two laterally placed the optic lobes. These are larger and these are also well developed. And these are the corpora. Another part it is called as a corpora crura cerebra, nervous tissue band, which is present in this, start from this region, crura cerebra, or in this side, crura cerebra, it is present. So this brain ventricle, this is the cerebellum, it is well developed. Hind brain part, it is called as a cerebellum and medulla oblongata. Cerebellum again divided into the parts. Middle part, these are transversely presence of this plate, these are called as the vermis, and these are again divided into the lateral lobe and flocculus. So this lateral lobe, lateral side, lateral lobe, and these are the smaller one, flocculus. So these are also related, highly developed, because of there is a instinctive behavior of these animals. Then these are the central portion, it is called as a vermis, lateral side, lateral lobe, and these are outgrowth like structure, these are the called as a flocculus. So the structure showing cerebellum, these are the white matter and gray matter, and this showing like this way, and this is called as a arbor vitae. This arrangement, it is called as a arbor vitae, which is present only in the mammal and birds. So this is the part, also olfactory bulb, largest part, cerebral hemisphere, pineal body is present, then also optic chasma, lower surface, and this is the cerebellum, well developed cerebellum. In between them there is a medulla oblongata, and lower surface there is a crura cerebra, nervous tissue band. This is the spinal cord. So this part, cavities, lateral ventricle, then there is a presence of, these are the optic lobes, then metacil, medulla oblongata cavity, it is called as the metacil. So this is the medulla oblongata, and this is the posterior commissures. So this is the presence of diencephalon in this side. So olfactory bulb, then lateral ventricle. The in this lateral there is a absence of rhinocele, only lateral ventricle, and presence of also foramen of Monro, diencephalon cavity. We see in the next diagram. Cavities in this also presence of all similar other animals or other vertebrates. So characters, it is larger than the reptiles and short and broad. Olfactory lobes are small or due to the poor sense of smell. Then two cerebral hemispheres, these are larger, smooth, and over diencephalon, and these two meet cerebellum. Pallium, or covering of this the cerebral hemisphere, it is called as a pallium. But in this animal, corpora striata is greatly enlarged and very well developed because of their aerial habitat. Then also third ventricle or diocese, it is narrow due to the development of optic thalami. Optic lobes are well developed in correlation with the keen sight, but they are somewhat laterally displaced. Cerebellum also enlarged, and several superficial floculi, folds, these are called floculi, and these are related to the muscular coordination, because aerial animals and equilibrium, maintain the body equilibrium, such as flight or perching. So brain bird, it is also well developed than the reptile brain. Then next one is the last one, mammals. In mammals, in practical, you study in the last year fowl brain. So this is the brain or rabbit brain. These are at the cerebral hemisphere, it goes number of convulsions. The olfactory lobes are also well developed, or olfactory bulbs are well developed. Forebrain part, olfactory bulb, cerebral hemisphere, and diencephalon. And in between them there is a longitudinal fissure is present. So in this brain consisting is fissure. This is the Sylvian fissure. Then also outer surface, median fissure. And part convulsions are called temporal lobe, occipital lobe. Then these are the dorsal surface, and ventrally these are showing renal fissure, hippocampal fissure. Then also these divided brain are into the many parts, or these are many convulsions showing in the mammal brain. In the diencephalon, in this side also, there is a cross of nerve, there is a presence of optic chiasma, then pituitary body, and also infundibulum. And in this mammal only, there is a presence of corpus albicans. These are hormonal control only related to the sex hormone. These are called as the corpus albicans, only found in the mammals, not other animals. And this also nervous tissue band, it is called as a pons varoli. It is also present only in the mammals. And this large cerebral hemisphere, laterally these are joined or connected by, it is called as a corpus callosum. Next diagram we see in this. So this is the part, olfactory bulb, cerebral hemisphere. This is the cerebellum. It is also similar. The cerebellum divided lateral lobe, flocculus, and vermis. This divided, and the cerebellum, there is a no cavity. Cerebral hemisphere cavity. In this also cavity. But this larger cerebral hemisphere connected by a transverse band, it is called as a corpus callosum. This is the part, it is called as a corpus callosum. So only present in the mammal. Other all animals, these are absent. Only presence of this pons varoli. This is the nervous tissue band. It is present only in the mammals, and also corpus albicans. It is also present only in the mammal. So these are the cavities of the brain: olfactory lobe, rhinocerebellum, lateral ventricle. These are the cavity of the cerebral hemisphere. Then diencephalon cavity, diocese. These together communicated together, it is called the foramen of Monro. Then diencephalon cavity, it is called as a diocese. Narrow canal in between them, there is a presence of ether. And medulla oblongata's cavity, it is called as a metasphere. And this is the structure, it is formed cerebellum, it is called as a arbor vitae, peculiar structure found only in the bird and mammal. It is called as a arbor vitae. So MCQ ask on this arbor vitae is present dash dash animal. So the answer is the mammal and bird. So this diagram showing, and in the also there are present optic lobes. In the mammal, optic lobes are 4 in number. So it is called as a corpora quadrigemina. Anterior and posterior, these are related to the acute hearing and also sense of sight. So these 4 in number optic lobes, these are called corpora quadrigemina. Because of 4 number, these are called quadrigemina. And next part, it is the hindbrain part. It is called as a cerebellum and medulla oblongata. So this is the part of cerebellum. Middle part, vermis, transverse partition. These are showing plates. It is called as a vermis. Then lateral side, lateral lobe, and there is a outgrowth like structure, flocculus. So brain is more advanced than the other all vertebrate. So this cerebellum, it is also more advanced than the other animals. So medulla oblongata also anteriorly broad, posteriorly narrow. And roof of this medulla oblongata, there is a presence of posterior choroid plexus. And this posterior, there are 2 choroid plexus. One is the anterior side and one is the posterior side. Anterior side, roof of diencephalon, there is a presence of choroid plexus. It is called as the anterior choroid plexus. This anterior choroid plexus and posterior choroid plexus produces cerebrospinal fluid. And this fluid is present in the brain ventricle or cavities, and it protects brain from the mechanical shocks. So this is the in mammal. These are divided prototheria, metatheria. Rabbit मतलब आपण बघूया आता फक्त. हा ब्रेन दाखवलेला आहे. हे आता डायग्राम मी दाखवले, कॅव्हिटी वगैरे सगळ्या ह्या दाखवलेल्या आहेत. कॅरेक्टरिस्टिक ऑफ द मॅमेलियन, 4 ऑप्टिक लोब्स, त्याला काय म्हणतात, कॉर्पोरा क्वाड्रिजेमिना. सेरेबेलम also large folded, median part is vermis, 2 lateral lobe आणि flacula. Mammalian hindbrain include pons varoli, transverse band of nerve fibers, and it connected cerebellum medulla oblongata with the midbrain. And these are also hindbrain contains center for the regulation of digestion, respiration and circulation. म्हणजे medulla oblongata चे फंक्शन आहे. दुसरा प्रेझेन्स आहे कॉर्पस अल्बिकन्स, कॉर्पस कॅलोजम. आता यामध्ये ह्या कंपॅरिझन दिलेले आहे बघा. ऐकताय ना, लक्ष आहे का? हॅलो.
येस मॅम.
हा, आता इथे कंपॅरिझन दिलेलं आहे बघा. डिव्हिजन पहिल्या याच्यामध्ये स्ट्रक्चर, फिश, एम्फिबिया, रेप्टाईल, पर्ड्स आणि मॅमल. आता काय काय सांगायचं इथं. पहिला पार्ट दिलेला आहे डिव्हिजन. किती पार्ट मध्ये ब्रेन डिव्हाइड होते? फोर ब्रेन, मिड ब्रेन, हाइंड ब्रेन. हे सगळ्यामध्ये सारखं आहे. फोर ब्रेन, मिड ब्रेन सगळ्यामध्ये सारखं आहे की नाही? इथं सेम सेम म्हटले. नंतर आता फोर ब्रेन कन्सिस्टिंग, फिश मध्ये काय काय आहे? ऑल्फॅक्टरी लोब आणि सेरेब्रल हेमिस्फियर नाही इथं. सेरेब्रम अनडिवायडेड आहे. ऑल्फॅक्टरी लोब आणि डायन्सिफर. यामध्ये सगळ्यामध्ये तसंच आहे. फोर ब्रेन कन्सिस्टिंग, सेरेब्रल हेमिस्फियर, ऑल्फॅक्टरी लोब अँड डायन्सिफर. मिड ब्रेन मध्ये काय आहे? ऑप्टिक लोब आणि क्रुरा सेरेब्रा. मिड ब्रेनच्या खाली नर्वस टिश्यू बँड असते, त्याला म्हणतात क्रुरा सेरेब्रा. हे सगळ्यामध्ये सारखंच आहे. नंतर हाइंड ब्रेन, सेरेबेलम, मेडुला ऑब्लांगेटा, हे पण सगळ्यामध्ये सारखंच आहे. आता बदल कुठं कुठं आहे बघा. ऑल्फॅक्टरी लोब विथ ऑल्फॅक्टरी पेडंकल इथं आहे. आता यामध्ये एम्फिबिया मध्ये पेडंकल नाही, ऑल्फॅक्टरी ट्रॅक्ट प्रेझेंट. इथं रेप्टाइल मध्ये पेडंकल आहे, बर्ड absent and in this also absent. Cerebrum single or undivided in fish. It is divided into the amphibia. सगळीकडे मग असा डिवायडेड आहे. मॅमल मध्ये मात्र मेनी कन्व्हल्शन्स आहे. लोबी इन्फेरियर्स and saxus vasculosus, these are present only in the fish, absent all the other vertebrates. Then hypophysis, infundibulum, these are present all the animals. Pineal stalk, pineal body, these are present all the animals. Only parietal body is present in the reptiles. Pineal body present in all vertebrates, but parietal only present in the reptiles. Then optic lobes, it is also called two optic lobes. These are called as a corpora bigemina. All animals there are four number, but only in the mammal these are 4. It is called as a quadragemina. Then crura cerebra present all the animals. Cerebellum enormously enlarged in the fish, then less developed in the amphibia, less developed in the reptiles, and less developed in the also birds, but it is well developed in the mammal. In the bird also well developed. Then pons varoli only present in the mammal. Medulla oblongata long in fish, short all animals or all vertebrates. Corpora restriformia, these are present only in the fish, absent all vertebrates. Corpora striata, it is present all the animals. Then corpora callosum, it is only present in the mammal. Two lateral cerebral hemisphere connected by a band, it is called as a corpus callosum. And corpus albicans, it is also present only in the mammal, absent other all animals. So this is the comparative account of brain related to the practical. अजून काय शंका आहे का तुमची? बोला आता. हॅलो.
येस मॅम.
हा, समजलं का?
येस मॅम.
किती जण आहेत बघा. काय शंका आहे का याच्याबद्दल? याच्यावरती तुम्हाला एमसीक्यू विचारतात, फुल क्वेश्चन विचारतात. हा इम्पॉर्टंट, खूप इम्पॉर्टंट टॉपिक आहे. कंपॅरिझन असेल फिश मॅमल, फिश, रेप्टाईल असे विचारले जातात. शंका काय आहे का? नसेल तर मी इथं थांबते आता. तुमचं प्रॅक्टिकल कारण नंतर आता माझ्याकडे जे 2 प्रॅक्टिकल होते, पहिले एक होतं ते हार्ट वरचं होतं, आताच आहे ते ब्रेनचं आहे. त्यामुळे माझ्याकडचे प्रॅक्टिकल आता संपलेले आहेत. तुम्हाला काय शंका असेल तर तुम्ही विचारा मला नंतर. सगळ्या ह्याच्या नोट्स पण मी तुम्हाला दिलेले आहेत. कुणाची काय शंका आहे का? बोला ना.
हॅलो मॅम.
हा,
मॅम सेन्स ऑर्गन आहे ना टॉपिक तुमचा, त्याच्यातले फक्त आईजच मिळालेत नोट्स, इयर्सचे नाही मिळाले.
त्याच्या नाही मिळाल्यात काय?
नाही,
तुम्ही
इयर
आता
नाही मिळाले.
प्रॅक्टिकलला इथे येत आहे ना, त्यावेळेला घेऊन जा.
ओके मॅम.
हा, बाकी सगळ्या मिळाल्यात का? वाचत चला.
हो
एक्झाम
मॅम.
एक्झाम आता फॉर्म भरायला लागलाय ना. एक्झाम तुमची एखाद्या वेळेस रिटर्न होणार आहे, त्यामुळे तुम्ही अभ्यास करत राहा. ओके, थँक्यू. मी थांबते आता इथं.
थँक्यू मॅम.
येस मॅम.
हा, वेलकम. कुठे गेले? इथे जाऊ.
हाईड करा नाही ते.
इथं
हे एक मिनिट सोडा.
स्टॉप शेअरिंग.
नाही नाही थांबा. जाऊ दे.